So guys, yesterday we looked at the extrinsic pathway of cell apoptosis, or basically apoptosis, which is programmed cell death. All right, and looking at these pathways, we looked at the extrinsic pathway, and we said that apart from the extrinsic pathway, there is an intrinsic pathway. Okay, so together, the extrinsic pathway and the intrinsic pathway, they form the initiation pathway or the initiation phase. Okay, so looked at the extrinsic pathway, and for us to do justice to the initiation phase, it's good for us to look at the intrinsic pathway too. So the intrinsic pathway of apoptosis will be having definition, and we'll be looking at the pathway from pictures. You could say beautiful pictures for you guys here. When we look at it, we'll also name some important substances that are actually found in this pathway. Okay, and by so doing, we must have done justice to it. So the introduction now. We said that intrinsic pathway, namely mitochondrial pathway, can be triggered by a number of factors or stresses. Okay, so this pathway can either be triggered by DNA damage, ischemia, or oxidative stress. Okay, and this can lead to what? Mitochondrial outer membrane permeabilization. That's the MOMP, mitochondrial outer membrane permeabilization. Okay, and the subsequent release of what? Pro-apoptotic mitochondrial proteins such as what? Cytochrome C. Okay, you can see this is the cytochrome C here, and the cytochrome C is the singly most important protease that is released from this pathway. Okay, so we'll be interrelating everything, and you guys get to see how the cytochrome C is released, the other genes that are also making this pathway a reality. Okay, so now guys, it is started by a release of cytochrome C from the mitochondria into the cytoplasm. You can see that from here, cytochrome C is released from the mitochondria into the cytoplasm. All right, anything you see here white is the cytoplasm. Okay, now the release of cytochrome C is controlled by what? Balance between pro and anti-apoptotic members of the BCL family of proteins. Okay, so now these are the, you see many genes here. Okay, they are the what? BCL family of proteins. Okay, now we have the anti-apoptotic. Those are the ones that are against what? Cell death. Then we have the pro-apoptotic. These are the ones that are basically what? Supporting cell death. Now the anti-apoptotic, we have the BCL2, BCLX, and the MCL1. Okay, it's a BCL2. I don't really have the MCL and the BCLX labeled. Okay, but you guys will see it here, and you can see that those are the anti-apoptotic, and you see that they are labeled with this red staff. Okay, tell you that they can inhibit the process. Okay, so now those are the anti-apoptotic. Then talking about the pro-apoptotic, we have the BAX and BAK. Okay, BAX and BAK. Okay, now regulated apoptosis initiators include what? The BEAM, the BEAD, the BAD, the PUMA, and the NOXA genes. Okay, so now these are what? Regulated by sensors of cellular stress and damage. Now, when upregulated and activated, they initiate apoptosis. Okay, so moving forward again, we said that the beam, BAD, and BEAD, they activate what? The BAX and BAK. Okay, and this is when the cells are what? Deprived of survival signals, when there's DNA damage, and when there's what? Misfolded proteins induce what? Endoplasmic reticulum stress. So looking at this now, the back, those are the pro-apoptotic. I will say that the regulated apoptotic, these ones will now activate this pro-apoptotic. Do you understand? This being bad, bead, puma, and noxa, okay, they will activate the backs and the back, because these are the ones that are what? Pro-apoptotic. All right, and these are in situations when maybe there's deprived of survival signals, when there's low chances of the cell surviving, and maybe there's DNA damage, there's misfolded proteins. Okay, and this can induce endoplasmic reticulum stress. Okay, So the BAX and the BAK, they'll form oligomers which insert into the mitochondrial membrane, and this will create channels which will allow proteins like Cytochrome C from the inner mitochondrial membrane to leak out. I told you guys that we'll be looking at the mechanism of this pathway. Okay, so now they also block the function of what? Proteins that are what? Against apoptosis, right? Those are the anti-apoptotic proteins, and we have the BCL2 and the BCLX, okay? And here we also have the MCL1, okay? And this is basically how this pathway is coordinated, and this will ultimately lead to what? Cytochrome C will now activate caspase 9. Caspase 9 will activate caspase 3, which is now at the execution pathway side. Okay, so now when you get to the execution pathway side, it will definitely lead to what? Apoptosis of the cell, right? So we have a video that will be coming on the execution and pathway. We'll be looking at the executional protein, which is the caspase 3. Okay, so you guys should stick to our YouTube channel because we have that coming. Right now, you can see where okay, I just want to connect something. All right, okay, these are the anti-apoptotic. Okay, now, okay. Oh, sorry, when there's survival signal, there will be no leakage of cytochrome. Okay, but now when there's decreased survival signals, it will cause leakage of cytochrome, and this cytochrome will activate what? Caspases, and it will definitely lead to what? Apoptosis, right? So guys, that's it about the intrinsic pathway of apoptosis. Basically talking about cytochrome C and how it activates caspase 9, and caspase 9 gets to activate caspase 3. Right, so see you guys.